Red blood cell don't have organelle and nucleus, just have hemoglobin proteins. But platelet don't have nucleus. But these type of blood cell have mitochondria, microtubular and actin cytoskeleton, glycogen granules, and some Golgi apparatus and ribosome. You can see in this picture microtubule, mitochondria, glycogen, and some granule, alpha granules, lambda granules, and delta granules, and some structure located into the surface or the central part of platelet. For example, dense tubular system like this, surface opening tubular, and here dense tubular system, the same this one. This is the ultrastructure of or morphology of platelet or thrombocyte. What is the function of platelet? The function of platelet is to prevent and stop bleeding. If a blood vessel is damaged or broken blood vessel wall, the body sends signals to platelet, which causes them to travel to the injured area. Once the platelet arrive at the site, they clump together to form a clot that helps stop bleeding. You can see here, blood vessel wall damaged. Here, blood vessel wall damaged, or broken blood vessel wall in this picture. You can see here red blood cell and here platelet. This is the situation of blood vessel is damaged. And in this picture, you can see activated platelet. Activated platelet start to secrete fibrin to form clots. Okay, to form clot to stop bleeding. You can see here clot to help stop bleeding. This one is platelet, activated platelet. Here is fibrin. All of these are clot formation. In this process, you can see the function of platelet to prevent and stop bleeding. Okay. There are three different types of granule into the platelet: alpha granules, delta granules, or dense body, or lambda granules. Alpha granules containing of secretory proteins, von Willebrand factor, and platelet-derived growth factor. Delta granules: calcium, serotonin, adenosine triphosphate, adenosine diphosphate, and pyrophosphate or pyrophosphate. Alpha granule involved into the formation of clot to prevent and stop bleeding. Alpha granule. Lambda granule as a lysosome, as an organelle for digesting intracellular component, for example, other organelle and some substance into the platelet, as a lysosome for digestive system into the cell. And this one, delta granule, calcium, serotonin, adenosine triphosphate, adenosine diphosphate, and pyrophosphate help to secrete or release another component of granule from platelet. For example, in prevent and stop bleeding, when all platelets forming the clot, prevent and stop bleeding. You can see in this table all the structure into the platelet. Surface opening tubule system, location hyaluromer, expedite rapid uptake and release of molecules from activated platelet. Dense tubular system, probably sequester. Calcium ion to prevent platelet thickness. Alpha granule contains fibrinogen, platelet-derived growth factor, platelet thromboplastin, thrombospodin, and coagulation factors. Two contained factor facilitated vessel repair, platelet aggregation, and coagulation of blood. Alpha granule involved into the clot formation to prevent and stop bleeding. And another type of granule, dense body, or this one, delta granule, consists of calcium, adenosine triphosphate, adenosine diphosphate, serotonin, histamine, and pyrophosphate. Containing factor facilitated platelet aggregation and adhesion, as well as vasoconstrictor. And lambda granule as a lysosome, containing enzyme, a clot resorption. In this table, you can see all structure and location and contents and function of all structure into the plant. Blood cell development. All blood cell originated from pluripotent stem cell. Okay, pluripotent stem cell. All blood cell. Pluripotent stem cell produce two types of multipotential stem cell: lymphoid stem cell and myeloid stem cell. Lymphoid stem cell produce lymphocyte. Myeloid stem cell produce another type of blood cell: platelet, erythrocyte, monocyte, eosinophil, basophil, and neutrophil. But lymphoid stem cell just can produce lymphocyte. But myeloid stem cell can produce other type of blood cells. Erythropoiesis. You can see in this picture poroerythroblast. You can see here a big cell have nucleus. This proerythroblast, the same chondroblast, the same fibroblast, the same osteoblast, okay, proerythroblast. This type of proerythroblast cell produce basophilic erythroblast. This cell have nucleus, multinucleate nucleus. The next step, basophilic erythroblast produce polychromatophilic erythroblast. Polychromatophilic erythroblast transform or turn into the orthochromatophilic erythroblast. The end of this stage, the nucleus come out, nucleus ejected, and these type of cell called reticulocyte. Okay, here site is mature type of cell, the same fibrocyte, the same chondrocyte, the same osteocyte. Okay, in this, all of this process take place or happen into the bone marrow. In this step, erythrocyte come into the blood flow. Okay, this is mature erythrocyte. By concave, don't have a nucleus and organelle, just have a hemoglobin. Sorry, you can see in this chart. Here is concentration, here is maturation. Okay, with maturation, the rate of hemoglobin increase, the rate of RNA decrease. Okay, because in this step there are a lot of ribosomes to form hemoglobin. Okay, there are a lot of ribosomes to form hemoglobin. When all ribosomes form hemoglobin, the rate of RNA decrease, the rate of hemoglobin increase. Okay, in this step, the rate of hemoglobin increase, the rate of RNA decrease. Or in this step, the rate of RNA increase. Okay, there are a lot of ribosomal RNA to form hemoglobin in this step. Okay, increase the rate of or the concentration of hemoglobin and decrease the concentration of RNA. This process about 6 to 10 days. The first step to produce mature erythrocyte. Leukopoiesis, this is erythropoiesis. For erythrocyte or red blood cell, this is leukopoiesis. For white blood cell, this is erythropoiesis, this is leukopoiesis. You can see here, as you know, all leukocytes originated from myelocyte except lymphocyte, because these type of cell originated from lymphoid stem cell. And another type of blood cell originated from myeloid cell. Erythrocyte, platelet, eosinophil, basophil, neutrophil, and monocyte. You can see in this picture progenitor cell or lymphoid stem cell, myeloblast, monoblast, myeloblast, promyelocyte, eosinophilic myelocyte, basophilic myelocyte, neutrophilic myelocyte, neutrophilic metamyelocyte, basophilic metamyelocyte, eosinophilic metamyelocyte, and this step, finally, you can see the mature eosinophil, basophil, neutrophil, and monocyte. I don't want in your exam to keep your mind all of this process, but you need to know